Hi again, guys. It's Kelly from the University of Florida, and I wanted to give you an update on your air potato fertilizer experiment. It's still me here and just showing you kind of what happened. These guys a little while to develop, so we're a little slower, but just as a reminder as to what you did, each class period had a separate treatment. Some of you guys were control, and you only put water in, and some of you guys had fertilizer. We then gave you some air potato plants, and then we gave you 10 beetles. And so you added the beetles to the plants, so that way the beetles would eat and do their thing. And then some of you guys watered, and some of you guys used fertilizer in the water, and you did that twice a week. And then after the beetles emerged, and that's kind of like when a butterfly emerges from a cocoon, they're doing a very similar thing. So after you guys did that, we counted them and to give you this data. So we're going to talk about that. We had maybe some issues along the way, but that's okay. That's normal. So here's just to kind of show you what we did. So these were your plants and these were your cages. And this was a good friend of ours from, he's actually from Australia, who came to help us with this project and help us count these things. So if I don't know that it'll play. I wanted to, but it won't. Okay, but there's a little video. So basically it shows what he did was that he was cutting off pieces of the plant and then just looking for larvae, looking for adults. If there were larvae or like the baby bugs, he put them in here into some fresh leaves so they could continue to develop because we wanted to get as many insects as we could for you guys. And give you the, oh, there we go. It's going to play the video for you. So that's what he's doing. He is taking that kind of piece by piece, looking for the little babies, looking for adults, and then again, babies would go in here so they can continue to eat so we could add adults that develop to that count. Or if there is an adult, then he puts it in this little thing right here. Okay, so this one had like 2 in it already. Okay, and so then we took from that, we took some data. So like a 7th period, for example, in Miss Barenborg's class, there were 24 alive, and they did find a couple of dead beetles, but they cataloged that for us. And then, like I said, all of those little larvae, little babies, they went in here for each class period, so that way they could eat and grow to be adult beetles. And we got a couple more that way. So again, we wanted to see how many total beetles we would get. Okay, so just to show you, if you didn't get a chance to see these, and these rolled up leaves is actually where we find the larvae. So some of these rolled up leaves would go in that container I was talked about. So these are like little babies, and they're just feeding and eating inside of a nice rolled leaf here. Okay, so that gave you some of the background just to kind of let you know what we did find per class period, so you can find your particular class period in this section. Is that 2nd period we found 7 alive, 3 dead. Those were controls. I grouped the controls together. 7th period we've got 2 classes together, and one of them had a lot going. And if you remember, I know it was a little while ago, but there was in the back of Miss Power's classroom, this plant had a lot of damage, and so that shows us it sustained a bunch of beetles. It had a whole bunch of beetles. Some of them did die, but that's okay. The other 7th period class had 5 beetles alive, 2 dead. And then our fertilizer groups, it was just depending on the class period, but sort of ranged about the same. So 4th period had about 7 beetles that were alive, 3 dead. 5th period you had 6, so did 6th period that were still alive at the end of the experiment. But 4 were dead for 5th period and 5 were dead for 6th period. Honestly, this isn't a huge difference. And there's not a whole lot of, like nobody's winning here. This is just to give us some data, and each of you guys separated out a treatment so we'd have different replicates. Okay, what I gave you here, now we could do a different way and we could go by average beetles, but I just wanted you to see kind of the difference. So I totaled up the number of alive beetles and the number of dead beetles, like just put the whole number together, so total beetles that we saw on those plants. So if you applied the fertilizer treatment, we only saw 31 beetles total between all the class periods. And then for the control, we saw 46, so we saw some more here. We could do it, just so you guys know, we could do an average of class periods, so we could say fertilizer treatment had an average of 6 beetles along all class periods, and then the controls would have had an average of about, I don't know, 10, 12 beetles. So we could do averages, and we usually do that. But because we're so limited, to show a greater difference, I just piled all of our data together so you could really see what, between everybody, what the controls and what the fertilizers look like. So just water and just for with the fertilizer, we seem to see that the controls actually gave us more beetles. So just a different way to see that. What I'm actually talking about here is what that data actually suggests. So this tells us that when we add fertilizer to the soil, it's actually not helping us to produce more beetles, which is actually what we want. We're trying to produce more beetles so they can go out there and eat more of these plants and of those invasive air potato vines. So we need to make more beetles. So if we're seeing here that adding fertilizer doesn't help us make more beetles that can go out and eat, then that would tell us we need to make some adjustments. Okay, so why doesn't the fertilizer help? Why didn't it work in this category? I don't know. It's possible that we over-fertilized or over-watered the plants. Plants are kind of particular. Sometimes they don't like it if we give them too much water or like too much fertilizer or too much nutrients, too much salt. If there's too much of something, it can mess them up. So it's possible that we gave them too much. Okay. And then, like I said, too many nutrients, too many different things can potentially harm the plant. So that may have been what happened, is that the addition of that fertilizer actually made it not so good of a food source for those beetles. So I just want to ask some general questions, and I'm trying to keep this short. I just wanted to show you kind of what we got all together so you could see those things. But as you're kind of closing out the school year and thinking about science, I just want to say, what would you personally test next time? So if we were to do this thing again, or if you were able to do it, what things would you look at? Remember that you have to have a control. So you have to have, like we had water and then we had fertilizer. So we have to have something to compare to. We could compare that fertilizer to just water by itself. So think about, like, what would you compare to each other? What would be your two treatments? Just start with two treatments. It gets trickier from there. And then or this particular experiment, would you change anything about it? Would you add more plants? Would you use more class periods? Would you give, 2 plants per treatment? What would you do differently? And so I just want you to kind of think about those things as we close this out and think about it. I will say that we also have this kind of issue that it's taken us a long time to get complete data for you guys because this isn't the beetles' best time of year. They're a little slow. So you could think of them like bears. They like to hibernate a little bit. They like to sleep. And so they're just now waking up. So they're a little slow to wake up. Are some of you guys slow to wake up in the morning? I know I am. So that took us a little while. So what would you change? Maybe is timing of the year. Would that have something to do with it? So again, think through some of those things. What would you change? What would you do differently to make this thing work? Or just to try and get something completely different anyway. Stuff to think about. So just final thing here, any of those questions that you ask or think through or talking about, like, I would do this versus this, that's how we do science. That's science. That's what we're doing. We're just doing it on a bigger scale. But it's the same thing. If you're thinking through it, then you're doing science, just the way that we would. So with that, I hope that you guys learned something from this. Again, sorry our beetles were a little sluggish on waking up, so it took us a while to get to you. But I hope you enjoy your summer so much and think science. Do some good stuff. Have fun, and we will, I'm sure, talk soon. It was good hanging out with you guys. Enjoy your summer. Thank you so much. Bye.